All right, so welcome to topic 4, the genetic blueprint and energy production assignments. So hopefully, we're continuing to catch up on, the lost time that we had due to the cyber attack. And so hopefully by the end of next week, we'll be pretty much caught up. So try to, just get through this, understand the materials as much as you can. And then in week 6, we're planning to do our midterm exams. So we need to, make sure that we're catching up on all that lost information. So if you're still stuck behind, if you're still, slugging it to turn in those topic 3 assignments, really take these next few days to try to catch up as quickly as possible, because you do have that midterm coming up in a couple weeks. So we just turned in topic 3 assignments. You should be getting those, the forums graded and all of that posted sometime today, so that you can prepare for your topic 3 quiz. So that is due this Sunday. So we're kind of, shortening the deadlines, or the time periods to catch up from our missed time. So pay attention to the calendars. So we have topic 3 assignments were just due yesterday. Use the feedback that I provide and help you prepare for your quiz. That's due this Sunday. Sunday, not a Wednesday, Sunday. So we're beginning to work through topic 4. And we have 4 assignments in topic 4. Plus a little introduction to a project we're going to be working on throughout the rest of the term. Also, just pay attention that the last assignment in topic 4 does require some extra time. So just, plan accordingly for that. And then you just submitted all of your topic 3 assignments. And remember that last lab that you did an experiment where you looked at the basically, diffusion, or you showed osmosis in the sucrose concentrations that were in the solutions, and then you put the bag of dialysis tubing filled with water and saw what happened, right? So you checked at the final weight. So do make sure that you go back into that assignment. Okay, so topic 3 S4, and look at what the data that other students have posted. So that way you can kind of average out your data for the concentration part of this lab with other students' data. So we can kind of, see if your data is, along the same lines as what other students were getting. And then you'll use that as well as the temperature data that we give you to help you construct your first lab report. So I talked about this lab report in the topic 3 video, and then I also have a complete video on it, so you can watch this video that walks you through exactly what you need to do to complete this lab report. So all the information and what order and so forth. So check out that video. And then this actual lab report is not due until February 11th. So, you don't have to rush to do it right this second. But I do suggest that you, at least this week, or the remainder of this week, try to start researching the background information for the introduction part. And then you just completed the lab, so it should be fresh in your mind. The methods part, so you're going to write a methods section in your lab report. Might as well do that while it's fresh in your brain. Okay, so I suggest you work on those 2 parts of that report now, and then, but the final project's not due until February 11th. Okay. What happened there? So our topic 3 summary by Dr. Blackwell, you can watch this video, gives you a good kind of summary of what we talked about in topic 3, and then an introduction to the material covered in topic 4. You have your Adigia assignment, so with your Adigia OpenStax textbook, you're going to review topic 4. And I kind of suggest that you do this first because this week there's some interesting parts, because we're going to be getting into something called cellular respiration, which can be a little tricky. So having some background information already in your brain will help you understand the assignments a little bit better. Your student assigned roles, I haven't posted as of this video, so you can ignore those names. They'll be up there when you look at it later. So let's take a look at our topic 4. So topic 4, we're going to get into nucleic acids. So, you know, we've already talked about the carbs, the lipids, the proteins, and the nucleic acids. So we're going to focus in on DNA and RNA, okay? We're also going to focus in on something called ATP. So those are kind of the 3 major molecules that we're going to be dealing with this week. So the first assignment that we're going to do is we're going to become familiar with DNA structure. So what is DNA? What is it made of? You know, what are the molecules that make it up, and all sorts of stuff. So you're going to start out by watching this video. And then once you kind of watch the video to familiarize yourself with the topic, then you'll dive into our workbook. So with the workbook, let me go back up here. You're just going to be answering the questions about, you know, what the structure of DNA is. And then we actually have these little pieces that you're going to cut out. So you'll go ahead and cut out these pieces and use them to help you kind of construct a little DNA molecule, learning the different parts that make up the DNA, okay? The rest of it's pretty self-explanatory. We'll get into how DNA is replicated, so how we take one strand of DNA and make another DNA molecule through something known as semi-conservative replication. Okay. And then we have some summary questions that kind of summarize everything. On page 120, you'll see these tables, so number 7. And I just want to clarify, when you are answering, posting your questions, everybody has a sequence that they have to write down from that table. So you can see like student 1, it's sequence 1, student 2, sequence 2. So what that's referring to is this table, and basically if you're student 1, you're going to be copying what you wrote in this first box. Okay, so this is sequence 1, this is sequence 2, sequence 3, sequence 4, sequence 5, and sequence 6. And what you're doing is you're taking that DNA sequence and you're simply writing the complementary strand to it. And this is DNA, so you know you're dealing with DNA, which you should only be dealing with the nucleotides found in DNA. So what would be complementary to the G there? Okay, G binds with what? C binds with what? And so forth. So you'll write that sequence that goes with the complementary strand that goes with this DNA sequence. And then for your assignment that you're posting, you're just posting your assigned sequence. Okay, so student 1, student 2, student 3, and so forth. Okay. And so once you've completed all of the questions in the workbook, go ahead and post your assigned questions in our discussion post. And then just be aware that the deadline for these assignments is a little, you know, we're still trying to play catch up. So it'll be Wednesday midnight this week. The second activity that you're going to be working through is, it can be a little tricky because it's cellular respiration. And cellular respiration is one of the more challenging topics in an intro to biology course. So we give you a video on it, but you know, it is a concept that you might just have to keep watching over and over again to kind of get it stuck in. And I highly suggest that there's an Amoeba Sisters video. So I mentioned the Amoeba Sisters when we had the cyber attacks, and that would be a good source if you can't reach Adigia. So the Amoeba Sisters video, they have a cellular respiration one. Also, Crash Course Biology is another good tool on YouTube. So they have a good video on cellular respiration as well. So those are 2 YouTube videos that I suggest that you look at if you're just not comprehending this whole cellular respiration process. So Amoeba Sisters and Crash Course Biology are 2 good sources for you. So once you've kind of gotten a little bit of background information, you know, check the Adigida textbook part as well as watch the videos, then you can start working on this activity. Okay, so get the foundation first before you kind of, mess with it there. And this is going to be an actual kind of simulation, and so you're going to need to prepare for the simulation by getting all these materials together. So you'll have to cut out pieces from your workbook, as well as extra pieces. So the extra materials are found here, and there's a lot of them. Okay, so look at all those pieces that you have to cut out. So if you have children at home, there you go. You can have them cut them out for you. Otherwise, just while you're watching TV one day, just, get some scissors and cut them out while you're, doing something mindless, okay? So once you have all those pieces cut out and you're ready to go, then you can actually go ahead and do the simulation. So you're gonna need to go through the simulation very carefully. So we have, the starting locations of the materials, so make sure your materials are where they're supposed to be. Now you don't have separate, you don't have two students working with you. So you could, just have one pile as student one pile for student two, or you can employ, your kids or your roommate or a sister, brother, father, spouse, whatever, to help you if you need to. And just, play the parts. So really work through this. And as you're working through, you'll notice there's little questions that you'll answer along the way. So answer the questions before you continue on. Okay, it's very key that you don't just, jump from here to here. So answer these questions first before you proceed to the next part. It'll make a lot more sense, okay? So you might have to run through this a couple times just to get the hang of it, because it can be a little confusing of, you have these little tokens and carbon and all that fun stuff. So, maybe run through it once or twice just to make sure that you're getting the questions completed and you've answered everything correctly. Once you've answered everything, then you can go ahead and transfer your answer to the discussion topic. Okay. And then the third assignment is, it's actually not very much in our notebook here. If you take a look, it's just this, it's 10 questions. Okay, so question 1 through 10. And before you actually answer these questions, you're going to be going through this little animation. And if it doesn't work by clicking here, you can open it up with this OPB, PBS link as well. So you're going to do the animation, or at least read the information, and then you're going to answer all those questions, those 10 questions, based on what you learned. And then as far as what you're going to post, you're going to post all 10 questions in your discussion topic. Okay, so questions 1 through 10 all get posted. All right. And then we have the last one. So the last one is another kind of experiment that we're going to be working on. So in this case, just like with the last experiment you did, we'll give you the data for one experiment, but you'll need to perform the other on your own. So the first experiment we will have done for you. So basically it's this aerobic respiration experiment, which if we take a look, is basically it's this part. So obviously you're not doing this at home, so you can kind of read through if you want, but you're not actually doing this experiment. The data for that is right here though. Okay, so this is what the data ended up being. So you'll kind of read through this, and then on page 139, this first chart is where you will write that data that we give you. Okay, so the P respiration data, take this information and then just put it in the chart. Okay, so that way you can kind of interpret it and everything. The second experiment you're actually going to do, okay? So this experiment is a fermentation experiment. But prior to that fermentation experiment, you need to kind of do some prep work. So, let me see. All right, so that, okay, so that doesn't apply because that's the germination of the seeds. Okay, here's the information for the fermentation experiment. So you're going to need to use the glucose and the starch solutions that are starch packets that are in your lab kit, and then you should see some test tubes, and I believe they give you balloons. I can't remember. I think it's balloons, and you're going to use these to help set up the experiment. Okay, so let's see. I can't remember off the top of my head everything that you'll need. Oh, and you'll need yeast. So if you haven't had a chance to go to the grocery store, just make a quick run to, you can just get it at the dollar store. You just need, a little packet of it for this experiment. So there's some setup that needs to be involved because you're going to be making a glucose solution from the glucose you're given, as well as a starch solution from the starch that you were given. You'll need to buy that yeast. Make sure it's fresh. If you have yeast sitting in your pantry and you don't remember the last time you used it, probably too old for this particular experiment. So you might want to try getting something, just one little packet is all you need. And you're going to be setting up the experiment. So basically what you're doing is you're going to be pouring the solution plus the yeast into a test tube. And I don't think you're inverting it. I can't remember. I think they give you balloons. I can't remember off the top of my head because I haven't actually physically seen the lab kit. But you're going to see how the yeast react. Okay, so the yeast are the organisms in this case. So the yeast are taking in the nutrients. In this case, they're taking in the glucose, or they're taking in the starch. And that's the nutrient that they're going to be using to undergo fermentation. And a byproduct of fermentation is carbon dioxide. So you'll see these little bubbles forming if you do it correctly. And if you're using a balloon attached to the test tube, then the bubbles will, kind of show that the balloon is inflating a bit. Okay? So just be aware of that. And then you can go ahead and write down which one you were assigned. So let me go back. So we have just the ones that you were assigned. So you're gonna, if you were assigned the yeast fermentation and water data, then you're just going to be posting that rate of fermentation, okay, for that one. If you were assigned the starch one, then there you go. You just write the results for that. And then that way, once you get other students' results, you can kind of fill it into this average class data table and summarize. And then from there, you're going to go ahead and make sure that you understood what happened with the aerobic respiration part, which we didn't do that experiment, so make sure you still understand it. There's a video, I think, of Tori kind of going over, how it was set up and everything. And then the second part was the fermentation part. So he kind of demonstrates that, and then you'll be conducting the experiment on your own and posting those results. Okay, so once you've answered all of those questions in our aerobic respiration and fermentation lab, then you'll go ahead and post your assigned answers right there with the discussion topic. Okay. And then the last thing that you're going to be doing for this week or this topic is the integration project selection assignment. So as I mentioned before, we're going to be starting on a project that will go through the remainder of the term. So right now, the only thing that you're doing is you're choosing a topic. That's it. You don't actually have to begin researching or anything. But we do want to kind of give you some background of this assignment that you're going to be working through. So it's going to be about genetic diseases, and so you're going to actually choose a genetic disease to learn more about. And in that genetic disease, you're going to be learning things like, what causes it, what's the biology behind it, what are the treatment options, and all sorts of stuff. And then towards the end of the term, you're going to compile all this information into like a PowerPoint presentation that you will share with the class. Now, you're not actually presenting it to the class as far as like you talking about it. You're just sharing your slides. And so we'll see what other students wrote, and you'll learn a little bit about different types of genetic diseases. So after you kind of get an idea of what a genetic disease is from the videos, you're going to go ahead and click on this link, Integration Project Handout. So for now, you don't need to, worry too much about it. You can learn, read a little bit about the information up here. But you can kind of ignore this part because this is what you're going to include in the actual presentation. But at the end here, there's a list of all the approved genetic diseases. So you're going to choose one of them. Now the catch is no two students can choose the same genetic disease to work on. So if you choose one of these, you gotta post your choice right away so that no other student can choose it. Okay, so first come, first serve. If you add to discussion and say, I want to do Tay-Sachs disease, which you're actually not allowed to do Tay-Sachs disease, but let's say you were, then you'll most likely get approved for it. If somebody, puts Tay-Sachs disease a day later than you did, then they're going to have to choose a different topic. So no two students can choose the same topic. So I would recommend looking through that list early and picking the one that looks most interesting to you. So some of you may have some familiarity with a genetic disease already and want to do that one because you already know a little bit about it, or you know somebody who's afflicted with it. Some of them just look kind of interesting, like albinism. So you can kind of choose. There's all sorts. If there's something on this that's not on this list that you already know about and you're interested in learning more about, let me know, email me, and I can see if it's something that will work in this type of assignment. So not all things can work. I've had students ask me about like chromosomal abnormalities, but that's a little different from genetic diseases in this case that we're focusing on. So we can't do something like Down syndrome or trisomy 18 or anything like that. Okay, so you can take a look at the list. If something's not on there that you're interested in, feel free to email me. And then once you have decided, post it here with your selection, and then hopefully you'll get it. Okay, so if you were the first to request it, then you'll get it. Okay, so all of this needs to be completed by this Wednesday, and then once it's completed, I'll get back to you, Thursday afternoon, and then you can use that to help you prepare for your topic 4 quiz that's due the Sunday. And then just remember, also on the same day as that topic 4 quiz is your lab report, your first lab report. So, you're still kind of working through to get everything done. In the meantime, if you have any questions, feel free to email me, and we can work through them together. All right, good luck, everyone.